Don't look at my slides. We still have 8 minutes.
I just had a visitor the other night.
Oh yeah, good times.
Just neighbors get worse and worse.
Yeah, I don't really know what happened.
Did they say sorry for barking?
No, but I saw one of the girls the next day, and she just like was too ashamed to make eye contact.
I saw you at the union yesterday.
Yeah. Yeah, did you go to that comedy night thing?
Yeah, what was it?
Did you perform?
Yeah.
Dang. I was tempted to go down there, but I was also trying to practice, so you know. I don't know, I was just like really anxious and like trying to be a person, but like I don't know. So pretty nervous, but excited for this to be over. No, I'm not gonna eat anything. I just mostly want to pretend like I'm not here.
A while ago.
Yeah, like a while ago.
Like how long ago?
Like over winter break.
Yeah. Okay. Then like my subconscious, I was like, something's different. But I didn't go any further than that. Are you ready? Are you excited?
Just like smile and nod at me the whole time. Otherwise, I'm gonna start crying.
I'm so excited. I mean, I'm so excited to learn about things I know absolutely nothing about.
Yeah, same.
So I told my friend Tennis Abby just before this, I was like, I'm going to botany after this thesis because I'm so excited to learn things I have no idea.
I wear my plant socks just to kind of like amp myself up, you know? So. Yeah, did I show you these socks?
What did they say?
They say plants get me. No, yeah, I just emailed Jill. You should be somewhere. That's it.
Okay.
Want me to open it?
No. Yeah, open it to the first slide. Presentation, I think is that one, right? Where's the presentation?
Go to slideshow.
Slideshow
From beginning.
from the beginning. All right. Don't look.
Hey! Yeah. Yes.
Yeah. That's good. That's what you want. You want to be on the center stage. All the things so no invitations means you don't play. You should just winner. No. You're going to get into NCAA. You're going to win the conference. You get it tomorrow. Yeah, but there's that. I know it's going to happen. You just watch me on cue. We will wait for all you four to get food and then we'll start. You can get some food. Oh, there's no more food. Wow. This may be the first time all the research is consumed. Everybody has to share. Take the ball, yes, take the ball. I can sell mine. Would you like to buy tacos for $5? Okay, let's go ahead and get started. It's really a pleasure of mine to introduce Abby in the defense presentation today. I'll start by saying that the project that Abby worked on for a thesis began long before Abby arrived. It started with a field trip that Shannon Elbeck and I did to the Wind River to collect some lichens. We had this idea about lichens being bioindicators of active nitrogen deposition and what cool things we could do with not only the modern collections but our bearing specimens. So Shannon and I got a little bit of money and went out to the Wind River Range for a week. And at the end of the week, after all the beer was gone, we realized that there's no way I could do this project justice. We would probably fail miserably. So we got some more funding and Abby on board to do this project, which was a fantastic decision for us because, as you'll see, she's done really, really great work. Abby has been absolutely a pleasure to work with. She's probably one of the most optimistic, really, that I've worked with in a long time. I mean, she definitely is an inspiration. She's always positive. Her electron energy state is always at a high level. She moves through processes, and she's unflappable. Like nothing seems to bother her in terms of challenges, and she just goes through this stuff. And let me just show you a picture of what is Abby's sort of typical state, her perfect state. She's smiling, she's in the field, she's doing what she loves to do. And she's just, unfathomable. Nothing interferes with this woman's doing stuff done in a positive attitude. Only one time did I see Abby start to kind of tighten up and be upset. And it was actually the same day that this photograph was taken when I was helping her set up her field plots. She drove her own car out to the field site, actually at Tetons. And as we were going over Teton Pass later this afternoon, the car started having some major difficulties. The automatic transmission failed, the clutch failed. We had to pull over the side of the road. That's the only time I have seen Abby look to have some distress in her face. And but even you can tell she's kind of smiling. It wasn't that big of a deal. So even this, in fact, I had to say, now, Abby, look like you're distressed about this situation. So anyway, that was the only time I've seen her not really just moving through something with a positive attitude. And it was all kind of staged. Well, that part wasn't staged, but I think the expression was staged. And so it's my pleasure to introduce Abby as she gives her talk. Both Shannon and I were co-advisors for Abby's project. Abby did her undergraduate work at Duke before she came here. Some of you may not know this, but Abby is moving on from this master's work, and she's joined the new EPSCoR micro project as a PhD student. That'll start tomorrow. So, Abby, take it away and let us know what you did.
All right, good. Thank you, Dave, for that wonderful introduction. So my thesis is to understand sources and spatial patterns of atmospheric nitrogen deposition using ion exchange resin collectors, which are pictured here, and lichens in the gritty Yellowstone ecosystem. And so biologically available reactive nitrogen increased threefold over the 20th century. This figure is just showing different sources that contribute to that. So we have biologically fixed nitrogen here in blue, and that stayed relatively constant. Then we have fossil fuels in purple, and that's kind of increased with development. But largely, the increase in total reactive nitrogen was due to the invention of the Haber-Bosch process, which takes inert nitrogen gas and hydrogen gas and turns it into ammonia, which can be used as fertilizers. And so this large increase in reactive nitrogen through combustion processes and volatilization processes makes it into the atmosphere and can be transported to more remote ecosystems. And it has a whole suite of ecological impacts. Largely, we have oxidized nitrogen and reduced nitrogen. And oxidized nitrogen is often deposited as nitric acid, so it contributes to soil acidification and water acidification in these ecosystems. And it also will leach ions out of the soil. And reduced nitrogen, when it gets deposited, can also be converted through nitrification and have these same kind of cascade of influences. And it also leads to altered plant communities through altered competitive relationships, particularly in these mountainous ecosystems, which tend to be nitrogen limited, and plants tend to have low capacity to adapt to increased nitrogen deposition through increased leaf nitrogen or through growth. And so these can drive losses in biodiversity in these ecosystems and also alterations to plant communities. And so this is kind of an overview of atmospheric nitrogen cycling. So down here at the bottom, these are your major sources. So we get reduced nitrogen from volatilization off of fertilizers and also from animal waste in agriculture. We get oxidized nitrogens from fossil fuel combustion. And then there's also natural sources of nitrogen in the atmosphere from forest fires and also from soil microbial processes. And these can be deposited both as wet and dry deposition. The gas can be taken up by plants, and the gas can also dissolve in precipitation that gets deposited. Typically, that will have a relatively localized impact. And similarly, for oxidized nitrogens, it's highly reactive in the atmosphere and will form nitric acid, which gets deposited again, mostly on a localized scale. But then shown up here on the top, these are atmospheric processes for longer distance transport of these nitrogen species. So for oxidized nitrogen, it can react with organic compounds to form these. It's a class of compounds that gets referred to as peroxyacetyl nitrates, and they're stable at cool temperatures. So when they get high in the atmosphere where it's colder, they can be transported over long distances. And then when there's mixing of air, they're brought down to areas where it's warmer, and they'll thermally decompose and nitrates will be deposited. And then for ammonia, it can react with nitrate to form ammonium nitrate, or also with sulfate to form ammonium sulfate, and that's an aerosol that has a longer lifetime in the atmosphere and can be transported over longer distances. And so different forms of nitrogen are important because over the past 20 years, largely due to regulations on emissions of nitrogen oxides, there have been shifts in the relative emissions of these different species of nitrogen. So here's a map just showing the percent change in ammonium ions and nitrate ions in deposition across the United States. And across the United States, there's been a decrease in nitrate deposition and largely an increase in ammonium deposition. And due to these different mechanisms that can transport these different species of nitrogen and potentially different impacts on ecosystems, it's really important to understand what sources of nitrogen are contributing to deposition in different areas. And so we can understand sources of nitrogen by looking at nitrogen isotope ratios. So nitrogen has two stable isotopes. The light isotope is 14N, which has 7 neutrons, and the heavy isotope is 15N, which has 8 neutrons. And so when we look back at what our sources of nitrogen emissions are, agriculture tends to have a really negative ratio because lighter isotopes preferentially will evaporate off of fertilizers or waste. And then for oxidized nitrogen, these nitrogen emission sources tend to reflect the fuel that's being combusted. So for power plants, ratios between +5 and +25 have been measured. And for vehicles, -12 to +15. And then again, the background sources tend to be largely influenced by soil microbial processes, so they tend to be more negative, -50 to -20. But that can also be brought up by biomass burning or also by lightning that fixes nitrogen. But one important thing to note is that all of these isotope ratios were measured right at the source. So like in a cornfield, or you put a filter over the exhaust pipe of a car and measure the isotope ratio. And transport processes in the atmosphere likely change these ratios. And the one process that might be significant here is that aerosol formation increase the isotope ratio, because the lighter isotopes tend to stay in the gas phase while heavier isotopes will form aerosols. So nationally, we have this atmospheric deposition monitoring network, largely because emissions of nitrogen cause aerosol formation, which decreases air quality, which can harm human health, and then also the nitrogen deposition can lead to acid rain and poor air quality or poor water quality and things like that. So people are pretty interested in these questions. However, these sites are pretty sparsely located across the country when you're looking more at impacts to actual ecosystems, and largely they're concentrated on the East Coast because that's where a lot of deposition, or where a lot of emissions are. And many of these sites are at lower elevations and not in remote areas, which are really the ecosystems that we're interested in looking at. And so to get a better idea of nitrogen deposition in these more remote ecosystems and at finer spatial scales, we decided to use lichens. And so shown here are the two lichen species that I use in this study, Litharia vulpina and Usnea lapponica. And lichens have been used as bioindicators for over a century because they acquire their nutrients out of the atmosphere, and they don't have a cuticle that allows them to concentrate nutrients or to exclude pollutants. So they are highly sensitive to those, and oftentimes their tissues reflect what the atmospheric conditions are. And so here's kind of an overview of lichen biology. So here on the left, you have sort of your typical healthy lichen. You have the algal symbiont, which photosynthesizes and fixes carbon and provides that to the fungal symbiont, which provides structure and protection. And so under situations of low nitrogen deposition, it likely acts as a fertilizer for the lichen. And so the lichen can invest more nitrogen in these algal symbionts, and those symbionts in turn photosynthesize more, provide more carbon to the fungal symbiont, and that leads to growth. And in those scenarios, we see low percent nitrogen in the lichens and a high carbon to nitrogen ratio, which indicates that they're maintaining that balance between the algal and fungal symbiont. But under situations of high nitrogen deposition, it leads to toxicity in the lichens. Oftentimes, growth will be limited by other factors besides just nitrogen, but the lichens don't have a way to control their nitrogen input, and they still use it to create more algal symbionts. But those end up shading each other out and blocking light, and also slowing diffusion of carbon dioxide into the cell, and the increased maintenance costs associated with those end up being harmful to the lichens because they're not actually photosynthesizing more. And so in those scenarios, we see higher percent nitrogen in the lichens and lower carbon to nitrogen ratios. And so that's kind of the mechanism through which lichens reflect atmospheric nitrogen. And so here, this was a field study done across the northwestern United States. And on the x-axis there, you have the concentration of nitrogen in the lichen. On the y-axis, you have nitrogen deposition. And you can see there's a positive relationship between the two, but also there's a lot of variation within that relationship. And that's likely because lichens are influenced by a lot of other factors besides just nitrogen deposition. For example, they need water to be metabolically active. Temperatures have to be above 0 degrees Celsius. They require light to photosynthesize, so differing amounts of light could influence how nitrogen is incorporated. Additionally, there have been studies that show that lichens preferentially will uptake ammonium over nitrate. And then additionally, the location of the lichen within a tree could influence how much nitrogen is actually delivered to that lichen. So for example, stem flow tends to have lower concentrations of ions than through flow. So lichens in the canopy might have higher percent nitrogen than those on the tree trunks. And then additionally, lichen delta 15N values reflect sources. So, and these relationships haven't quite been as well quantitatively developed as the percent nitrogen. But here on top there are lichens that were collected along a major highway in California. And they tend to have a higher isotope ratio, which reflects combustion sources from vehicles. Whereas these lichens along the bottom that were collected along a remote Forest Service road in the National Forest in the Sierra Nevadas had a lower isotope ratio, probably more indicative of background ratios that we would expect. And then over here on the right, this is showing with increasing loading of nitrogen and fertilizers, isotope ratios in the lichens decrease. And so likely we think that isotope ratios in lichens that are reflecting combustion sources are higher, probably -5 to +10. And then lichen isotope ratios from agricultural sources or background sources tend to be lower, likely around -14 to -8. And so lichens really record these fine scale patterns that are really difficult to get from modeling and would be really difficult to actually put out collectors or get enough monitoring sites to understand these patterns. And so this figure is showing that on the left, this is a climate and air transport model where they've modeled deposition. And model deposition is pretty similar throughout the region. And then over on the right, these are nitrogen deposition values extrapolated from lichen nitrogen values. And you can see that even over these really small spatial scales, there's a large amount of variation within the lichen tissues. And so oftentimes when we model nitrogen deposition on these fine or on these smaller spatial scales, they're biased by a small number of measurements, and oftentimes we make assumptions that deposition patterns are driven by precipitation or elevation patterns and distance to emission sources. So here, this is Rocky Mountain National Park, and they modeled deposition based on the fact that emissions likely come from Denver to the southeast. And as air masses move up into the mountains, likely there's more precipitation and nitrogen gets deposited in that precipitation. And then as air masses move on to the west side of the Continental Divide, there's not a whole lot of nitrogen left. And that could be a good assumption, but also, I mean, there's not a whole lot of data in the northern part of Rocky Mountain National Park. And so oftentimes we're forced to extrapolate few measurements over large spatial areas. So maybe using lichens and actually getting some kind of data in these areas would be better than just this. And so we did this study in the Greater Yellowstone ecosystem, which is the largest protected area in the continental United States, and it's protected under the Clean Air Act. And the topography here is quite complex. There's several mountain ranges that cut through. So it's really interesting to look at these relationships between nitrogen deposition and precipitation or elevation gradients. And additionally, a lot of the land managers in this area are concerned about increased deposition due to agriculture that is just all over Idaho here in the Snake River Plain. And so, largely in this region as well, deposition is modeled as a function of precipitation and distance to emission sources. So here on the left, this is a precipitation map of the Greater Yellowstone area with highest precipitation in the Tetons. And that's reflected in the deposition model where you have higher deposition in the Tetons where precipitation is high and it's close to the source to the west of agriculture in the Snake River Plain. So then this is another study that modeled just an average deposition rate for Grand Teton National Park and looked at the spatial contributions of emission sources. And so these colors, the hotter colors, are showing those areas are contributing more to deposition in Grand Teton, and the lighter colors indicate those are contributing less. And what they found was that about 50% of deposition in Grand Teton originates from within 400 kilometers of the park, and 50% originates from areas that are outside of 400 kilometers of the park. And so this study really suggested that long distance transport is quite important, particularly in the Greater Yellowstone ecosystem, because the Yellowstone Plateau is raised, and they said it causes downwelling and mixing of air, which allows for high rates of long distance transported N to be deposited here. And then they also looked at the breakdown of different sources. And so their model suggested about half of the nitrogen in Grand Teton came from these agricultural sources. And then actually about a third comes from surface emissions of NOx, which refers to vehicle and other non-point sources of nitrogen. So this led us to really try to understand what the drivers are of deposition in this region. So the questions we asked: to what extent are these patterns driven by precipitation and proximity to emission sources? What are the major sources of nitrogen deposition across the Greater Yellowstone? And then how do lichens record nitrogen deposition, and how is that mediated by these other climate and environmental variables? And so what we did was we set up these 3 elevation transects at Jackson Hole, Grand Targhee, and Avalanche Peak to try to understand these relationships between elevation and deposition and distance sources. And so at each of those sites in those transects, we put out these ion exchange resin collectors, and we collected lichens and compared those. And then we also had this 4th transect that had increasing distance from the Snake River Plain. And that was to try to understand whether deposition decreases with distance from what land managers think is the major deposition source, or what kind of pattern we see there. And then in addition to these sites, we also collected lichens at other sites throughout the area to increase our spatial understanding of these nitrogen deposition patterns. And we compared those to climate model data and emissions data to try to understand how deposition and climate interact to create these lichen percent nitrogen values. And so at each of these sites, we put out 3 collectors that were out in the open and 6 collectors that were under the canopy. And this is because these collectors that are out in the open are probably a better indicator or a better measure of actual deposition, while the collectors that are under the canopy likely are better correlated to the lichen, what the lichen are recording. But oftentimes with the through-fall collectors, you get more variable measures of deposition and oftentimes higher measures of deposition because the canopy collects dry deposition and then kind of funnels it all down through the canopy. And so this is the collectors that we put out, and basically it's a funnel that will collect dry deposition and precipitation and funnel it down through a PVC pipe that's filled with this mixed bed ion exchange resin. And basically what that means is that there's little beads in there that have positive and negative charges. So when precipitation goes through, the ions in the precipitation will bind to those beads, and the water will drain out. And you can take these back to the lab and extract them with potassium chloride, measure the ion concentrations, and extrapolate using the volume of the extracts in the area of the funnel to understand rates of deposition over that area. And then also on those extracts, we measured isotope ratios. So for the nitrate isotopes, we used this microbial denitrification method. Huge shout out to Kyle who did all the troubleshooting with that. Basically what it does is it uses microbes to convert nitrate to N2O gas, and then that can be injected and the isotope ratios can be measured on a mass spectrometer. And then for the ammonium isotopes, we measure them using this procedure that's depicted here, where basically you put your sample into a bottle and you add magnesium oxide and seal it, and the magnesium oxide facilitates the conversion of the ammonium ions to ammonia gas, which diffuses into the headspace. And then you have a little filter pack there that has sulfuric acid on it, and the ammonia will react with the sulfur to form ammonium sulfate particles that are collected on that filter. And those are what are allowed to stabilize for 2 weeks to try to ensure that there's not fractionation of ammonia there, or of the nitrogen isotopes in the ammonia. And then those filters are combusted and the ratios are measured using a mass spectrometer. And then, so finally at each of the sites where we had ion exchange collectors, we had co-collections of lichens with the collectors. And up on the right, that's the Theria that we collected. And then here on this tree, the lighter green lichen is Usnea. And so this is some of the results. So here this is showing across those 3 elevation transects what the ion exchange resin collector measured nitrogen deposition across elevation. And so kind of one of the first things to notice is that these collectors that were under the canopy higher deposition, higher measured deposition than the collectors that were out in the open. And so that supports the idea that the canopies are collecting dry deposition and funneling it down. But across these two transects at Grand Target and Jackson Hole, largely we don't see a relationship with elevation. And so this suggests that deposition at these sites are really more probably driven by emissions, and high concentrations of ammonia gas have been measured at some of these sites in Grand Targhee. And so likely there's just this huge source of ammonia just to the west of those sites. And so it doesn't really matter how much precipitation there is, there's dry and wet deposition occurring, and you're getting relatively high rates of deposition for these sites, kind of regardless of elevation. But then at this transect out in Avalanche Peak, we do see this increase from the lower sites to the higher elevation site. And overall, the deposition at this site is relatively high, considering that there aren't a whole lot of anthropogenic sources of nitrogen directly near that area. And so this suggests that there is long distance transport. And particularly with the location of this site up near the peak, at Avalanche Peak, that could contribute to increased rates of dry deposition, particularly of aerosols, which other studies have found that on more exposed ridges or peaks, that facilitates higher rates of dry deposition just due to wind patterns coming up over the peaks. And then this is the transect with increasing distance from the Snake River Plain. And so here we expect to see high rates of deposition further west, closer to agriculture in the Snake River Plain, and then decreasing, which is not what we saw. There's pretty constant rate of deposition across that transect, which again suggests that long distance transport of nitrogen is actually pretty important in contributing nitrogen deposition to these ecosystems. And we can't just assume that these further east areas in Yellowstone are experiencing less deposition, which is kind of what the null hypothesis has been. So then these are those same transects and the lichen percent nitrogen values. And so largely the lichen percent nitrogen values, well, first of all, Usnea tended to have a higher percent nitrogen than Lithria. And that makes sense because Usnea is often classified as a more sensitive lichen species, and it's more impacted by inputs of nitrogen. And so we see that here. But largely across the Grand Targhee, Jackson Hole, and the distance transect, these are pretty similar to the ion exchange resin collector trends, where we get a little bit of an increase with elevation, but it's not significant, and it tends to be pretty constant across those elevation gradients. There could be higher incorporation of nitrogen at some of these higher elevation sites just because they tend to be wetter or have high relative humidity, which we think allows lichens to be metabolically active more and potentially incorporate more nitrogen. This Avalanche Peak site is not at all similar to the collectors. And I guess the first thing to notice here is that at that highest elevation site, Usnea wasn't present, and so that could indicate that there are high levels of deposition that are excluding Usnea from existing there. Other than that, I mean, this pattern could be because there are other environmental variables that are influencing how lichens are incorporating nitrogen. Those areas in the east part of the park tend to be a little bit colder and drier, so maybe that's changing the trends that we observe. But that's a little bit of a puzzle. And then when we look at the correlation between the lichen percent nitrogen and the collector nitrogen, we see this positive relationship between the two, but it's a pretty weak relationship, which suggests that other factors besides just nitrogen deposition really influence lichen percent nitrogen. And so oftentimes, even within the same site, lichen collections could vary due to the location of the tree, or they were collected on different tree species, or potentially canopy density is altering the amount of dry deposition that's reflected in lichens. So there's a lot of different kind of micro habitat factors that could influence what lichen are actually recording. And then this is showing the lichen delta 15N values. That dashed line is at -8 per mil. And so values below that suggest those are reflecting agricultural or background emission sources, and above that suggest fossil fuel combustion sources. And so here, largely the lichens indicate agricultural sources, except for a few of these sites at Jackson Hole, which I'm going to come back to talk about the isotope ratios from the collectors. And so these are the isotope ratios of the two different ions that were measured in the collectors. So the squares are nitrate ions, and the diamonds are ammonium ions. And the two different colors represent collectors that were on the open are orange, and collectors that are under the canopy are blue. And for these, there's just one measurement at each site because these measurements were pretty difficult and intensive to do, and we didn't expect sources to change within a site. But probably the first thing you notice here is that all of these isotope values are a lot higher than those reported in the lichens. And that could be because isotope values are raised due to these transport processes. And maybe lichens are discriminating against heavier isotopes when they have such high inputs of nitrogen. And so you see the lower isotope ratios. Additionally, maybe lichens are preferentially taking up ammonia gas or something like that has a smaller spatial scale and isn't as influenced by transport processes. And then another thing to notice is that across all the sites, the isotope values measured in the collectors out in the open under the canopy weren't different. And several studies have shown that tree canopies can process nitrogen, uptake nitrogen, or leach nitrogen. But since there weren't differences, that suggests that the canopies weren't really doing that. And then when we look at the site at Jackson Hole, which some of those lichen values also had high isotope ratios, and also the transect across the Snake River Plain, we see some really high isotope ratios. And so that suggests that aerosol formation and aerosol deposition likely is contributing at these sites because aerosol formation tends to raise those isotope ratios. It could also just be, particularly for the oxidized nitrogen, so the nitrate species, it could just be from combustion sources that have higher isotope ratios. But this kind of supports the idea that long distance transport is really raising nitrogen deposition across the greater Yellowstone system. And so then here, these are lichen collections at all those sites that we collected at across the greater Yellowstone ecosystem. And so the kind of cooler colors, blues and greens, indicate lower percent nitrogen, oranges and red indicate higher percent nitrogen. And the goal with this is to create kind of a map of deposition hotspots. But because there's so much variation over small spatial scales, it's really, it wouldn't really make sense to interpolate between those points. And yeah, so this, okay. And so then additionally what we did was we compared these lichen percent nitrogen values with oxidized and reduced nitrogen emissions over distances that range from 0 to 800 kilometers and different climate data. And we didn't find a whole lot of correlations, which suggests that these lichens are more responsive to these really fine scale patterns that we're not measuring, in that these more regional patterns, deposition, emissions, and climate are more secondary. And so going forward with this, so going forward with this, I think it's really important to think about kind of the scales that lichen are useful at. And if we start averaging some of these lichen values over larger areas, maybe if that's more reflective of emission sources, or if lichens are just really only recording these really fine scale patterns. And then to kind of follow that up, we also looked at the lichen carbon to nitrogen ratios, which are likely a better measure of the impact of deposition on these ecosystems because they show whether or not the lichens are able to respond to and actually utilize that nitrogen for growth. And so here the low ratios in reds and oranges represent stress to the lichens, and then the greens and blues represent higher ratios, and those are lichens that aren't as stressed. And so largely what this is showing is that kind of across the greater Yellowstone ecosystem, the current rates of nitrogen deposition are creating stress in these lichens, and they're actually probably influencing the ecosystems. And so in conclusion, in these areas that have really well-defined sources and environmental gradients, and deposition patterns do reflect sort of those environmental gradients and emission sources. But in areas like Northeast Yellowstone that don't have these well-defined sources, it's a lot more difficult to model those deposition patterns, and it's difficult to know what the long-distance transport sources are, how those get deposited on the landscape. And then the isotope ratios largely suggest agricultural sources, but also there's probably a significant contribution from long-range transport of nitrogen. And then finally, the lichen tissues do reflect nitrogen, but that signal is pretty mediated by other factors that are really difficult to measure. And so really getting a better understanding of how lichen biology is influenced by these different environmental factors will help us to better understand what these spatial patterns of lichen percent nitrogen are telling us, and also potentially help us to better understand these herbaria collections of lichens that we have that can help us to understand trends over time. So with that, I'd like to thank Shannon and Dave for their endless enthusiasm and support and feedback, and Ursula, who's also on my committee, for joining late and just being super flexible. And then also thanks to Jill McMurray and Dave Evans, who are our collaborators on this. Shout out to the Williams Lab, Jason, Will, Jimin, for just always being willing to help out in the field and provide feedback. And then Greater Yellowstone Coordinating Committee, Dub, NPS Research Station, and the Wyoming NASA Space Grant all provided funding. And then huge thanks to Stable Isotope Facility, Al, and the Geochemistry Lab that helped with analyses. And also huge thanks to David Mayer, who's an undergrad and helped me with all this field work, hiking up these elevation transects with a bunch of equipment and climbing trees to get to lichens. So with that, I'll take questions. Pretty much from June to September. Yeah, so we didn't extract values throughout the year because they probably do change from summer to winter. And also, lichens probably also are only incorporating nitrogen for wood growth or some subset of the year. Yeah, what was the question?
Oh, just you had a subset beginning in September. I was wondering if it was a full count.
Yeah.
You think it would have been different if you also counted snow?
Different in terms of
Amount of nitrogen might be covered.
yeah, so I think this area deposition is largely driven by agricultural sources. So that's likely to be a lot lower temperature. but at the point where long distance transport is also contributing, but we're not really sure how much and what those sources are exactly. Those could be higher in winter because you get more combustion of fuels for heating and things like that. So definitely a shift between seasons, but not exactly what
So, you introduced a lot of other potential factors that issues in the were you able to quantify any of those factors and incorporate those into your knowledge?
Yeah, so that's definitely something that we're looking at. It's really hard to, I think, quantify these climate variables on the scale that lichens are acting. And so we looked at correlations between precipitation, or days of precipitation, relative humidity, different temperature measures, and there aren't really good correlations with lichens. And I think that's just because even on the same tree, different lichens on that tree can experience different really microhabitat environments. So yeah, it's definitely a future direction that difficult to address.
A lot of lichens have not just health and we also have different bacteria. In fact, those of you in New Zealand have found that some bacteria as well. And so another interesting tangent would be to look at, the effect of nitrogen deposition on the composition of algal seed inside. And I can see go back to the varying from 5 to now and see if there was a change. The nitrogen deposition is having a big effect on lichens in the earth. These look nice and healthy, but it doesn't necessarily mean there hasn't been a change. And it's pretty easy to do. Low hanging fruit, low
Thought seriously about this. We've been consulting with some folks. They're suggesting that the quality of these are so we thought a lot about doing exactly that. But dissuaded from that. You don't necessarily need those sequences. You can probably use mix or something that would be still
Alex.
Fires up here?
There were a lot of fires, yeah. Yeah. So I think that brings up a couple of good points, actually. So first off, fires likely contribute a lot of organic nitrogen compounds, which are really difficult to measure. And so they largely just get ignored. But there have been some studies that suggest that up to 20% of deposition could be in those organic forms. But also, lichens are obviously incorporating nitrogen over really long time scales. And so, I guess in response to the how are fires affecting lichen nitrogen, a large area of the Yellowstone has been burned, so hopefully that kind of evens out. But yeah, we don't really know how fires affect them.
How well the atmosphere versus
Another great question. And yeah. Yeah, and so I definitely do think that some of the higher rates of deposition that we saw near Jackson Hole are because of some of these inversion processes. In terms of mixing of the atmosphere out near Avalanche Peak, though, that sort of region of Yellowstone, I think largely has been thought to be kind of protected because it is more remote, and so there hasn't been a lot of work that has looked at deposition or atmospheric patterns in that area. I mean, largely because they're ubiquitous throughout the greater Yellowstone. And so out of convenience, the knowledge that we need to find them in most places.
The rest of the group